Charles Darwin proposed the theory of natural selection. Many people at the time did not accept his theory. There was a different theory at the same time as Darwin's theory. The different theory said that changes in an organism during its life could be inherited. Who proposed this theory? Well, this was created by John Baptist Lamarck. He was the predecessor to Darwin, who had these views. Studying fossils helps scientists to understand how living things have evolved. Figure 1 shows a fossilized snake. Now looking at Figure 1, we can see that of the snake, only the bones remain. And we can clearly see here the backbone alongside the rib cages of this snake. Now, the first part of the question asks us to explain how the fossil in Figure 1 may have formed. Now it's a 3-mark question, so we need to make 3 clear points. Now, a fossil forms when, first of all, an organism is covered in sediment or mud. So that's our first point. The dead snake was covered by sediment. Now following this, microorganisms in the mud are able to decompose much of the snake, but they cannot decompose the bones, only the soft tissue such as muscles and skin. So this will be our second point, that the soft tissue decays, but the bone does not. Now over time, more and more layers of sediment will pile on top of this organisms, and with this will come high amounts of pressure. Now this pressure will force minerals from the soil into the bones, and this mineral will replace the bones, thus creating the solid structure that is a fossil. So this is our final point, that because the bone is not decayed, it is replaced by minerals, thus creating a fossil. There are many types of rat snake in the world. Figure 1 shows 2 types of rat snake. Let's have a look at Table 1. Well, first of all, we can see the Japanese rat snake, which lives in a grass area and is green. Then we also have the Texas rat snake, which lives in a dry and dusty environment and is pale brown. Let's have a look at this part of the question. The different types of rat snakes have evolved from similar ancestors. The rat snakes have evolved to suit their environments. Explain how the Japanese rat snake evolved to be different from the Texas rat snake. So this is a 4-mark explain question. Now, to get to level 2, that is 3 or 4 marks out of 4, our answer needs to be detailed and coherent. And not only this, it must make logical links. So, now, the question mentions that the rat snakes have evolved. So let's recap what evolution is. Well, first of all, there has to be natural variation in the population. In this case, that variation would be to do with colour, since the differences between the 2 snakes are their colours. Secondly, some individuals will be better adapted. That is, some individuals will have a colour which makes them more suited to their environment. Thirdly, the better adapted individuals are more likely to survive and reproduce. So, as we said before, looking at the table, the Japanese rat snake is green and lives in a green environment due to the grass. So we need to describe to the examiner how these snakes became green. Well, as we previously mentioned, we need to first state that initially there's lots of different colours of snake. Now, since we need to be detailed and coherent, we want to go into more detail than this and mention what causes this. Well, these colours are determined by the genes of the snakes. So this will be our second point. Next, we could say that some of these snakes are closer in colour to the green of the grass. Now, these snakes will have an advantage since they will be better camouflaged. This is the explanation for our statement. Now, what is the result of this? Well, the green snakes are more likely to survive, and you could include here the reason why, such as they are less likely to be eaten by predators, and they're more likely to survive, so therefore they're also more likely to survive and reproduce, or breed. Once again, to make our answer more detailed, we're going to link it back to the genes. So, if these individuals are more likely to reproduce, so the survivor's offspring will inherit these genes, which give them the advantage of being green, in all likelihood. It's important to remember that this will occur over a very long timescale. So once repeated over many generations, eventually all the rat snakes in Japan became green. Many species of snake have become extinct. Give one reason why a species might become extinct. Well, the extinction of a species is when there are none left of that species due to mass death. Now this could be caused by a multitude of factors, and, for example, new predator arriving, which then eats many of the individuals until none are left. Alternatively, we could have said changed to the surroundings. This could have been due to a change in the temperature, which the species was not adapted to, for example. It could be due to the migration of new diseases to the area. Alternatively, it could have been due to new competitors, so that there becomes insufficient food for this species. Or finally, due to a catastrophic event, such as a mass drought or a volcanic eruption, which wipes out many of the individuals from many species in an area. A gardener wants to add compost to the soil to increase his yield of strawberries. A gardener wants to make his own compost. An airtight compost heap causes anaerobic decay. Explain why the gardener might be against producing compost using this method. Now this is a 2-mark explain question, so you want to make both a statement and also give reasoning behind it. So let's think about what happens when anaerobic decay occurs. Well, in this process, methane is produced. Now, this is our first point, the statement, that anaerobic decay produces methane. But why is this a problem? Well, the reason this is an issue is that methane is a greenhouse gas. So that's our second point, and the reasoning behind it. Now, a greenhouse gas means that if this is the Earth and the atmosphere, methane in the atmosphere allows the sun's rays to pass through to the surface of the Earth, but any radiation that's emitted from the Earth's surface is reflected back. In this way, it acts in a similar way to a greenhouse, increasing the temperature of the Earth, thus causing climate change. The gardener finds this research on the internet. A carbon to nitrogen ratio of 25:1 will produce fertile compost. Look at Table 2. Well, Table 2 shows the type of material to compost, which is chicken, horse and peat manure, the mass of carbon and the mass of nitrogen in the sample, and from this, the carbon to nitrogen ratio has been calculated. Now, we are asked to determine the ratio X in Table 2. Now, as you can see, the other two ratios are given with a number to 1. So to do this, we need to divide the mass of carbon and nitrogen in the peat moss by the mass of nitrogen, since dividing any number by itself will give you an answer of 1. So our calculation here will be 9.8 divided by 0.2 to find the ratio of carbon to nitrogen. So this gives us an answer of 49. So our final ratio will be 49:1. Which type of material in Table 2 would be best for the gardener to use to make his compost? Justify your answer. Well, looking back at the table, we can see that just before it states that a fertile compost is that which has a ratio of carbon to nitrogen of 25 to 1. Well, as we worked out from the previous part of the question, the carbon to nitrogen ratio of peat moss is 49 to 1. So, looking at the three possible types of materials, we can see that horse manure has a ratio closest to 25 to 1. So this will be our answer. Horse manure. And the reasoning, as we've just said, is that it's because it has the closest ratio to 25 to 1. Some of the leaves from the gardener's strawberry plant die. The dead leaves fall off the strawberry plant onto the ground. The carbon in the dead leaves is recycled through the carbon cycle. Explain how the carbon is recycled into the growth of new leaves. This question is a 6-mark question, and it's level marked. Now, to get to the top band, that is level 3, for 5 or 6 marks, your answer needs to be detailed and coherent, and logical links must be made. Now, it's referring to the carbon cycle here, but we need to make sure we're specific to the example given of the strawberry leaves, as opposed to just generally stating the carbon cycle. Well, let's have a think about what happens. Well, when leaves die, they fall to the ground, as it states here, and microorganisms, known as decomposers, will digest this leaf. In the process, the microorganisms respire, which is a process that releases CO2. Let's write about this part of the process before continuing. Well, first of all, carbon compounds in the leaves are broken down by microorganisms. These respire, releasing CO2 into the atmosphere. Now, following this, the gaseous CO2 is in the atmosphere. This can then be absorbed by plants. Now, plants use CO2 for the process of photosynthesis, which is the way in which they produce sugars in the form of glucose. These will be our next points. That plants take up CO2 and use it in the process of photosynthesis. This converts CO2 into glucose. Now, the glucose produced is a source of energy, and it's used to make proteins. Now, these are essential for growth, so this links it back to the growth of new leaves. Alternatively here, you could have mentioned amino acids or DNA in the place of proteins. So, looking back our answer, we can be sure that we have both statements, as highlighted here, and explanations. Now, since these are present throughout our whole answer, we can be sure that it is both detailed and coherent, and has logical links. Figure 2 shows 2 strawberries. Both strawberries were picked from the same strawberry plant. Both strawberries were picked 3 days ago. The strawberries were stored in different conditions. Well, looking at figure 2, we can see that strawberry A has a large patch of mould on it, and looks generally more withered than strawberry B, which is in perfect condition. The question asks us to give 3 possible reasons that may have caused strawberry A to decay. Well, as it states in the question, many factors are controlled, since they are from the same plant and were picked at the same time. Therefore, the only thing that could cause the difference in the rate of decay were the conditions. We can see on strawberry A that there is mould, as we've already said. Now, this requires microorganisms to create the mould, and for them to digest and decompose the strawberry, it requires warmth, oxygen, and moisture. So, the conditions in which strawberry A were kept must have allowed the growth of mould by providing these factors. So our first point will be that it was stored at higher temperature. Next, we can say that it is stored in conditions with more oxygen, since oxygen is required for decay. Next, we could say that it is stored in a place with more moisture than strawberry B. And you could also have said, instead of one of these points, that the storage conditions contained more microorganisms that cause decay. Many different types of animals are produced using selective breeding. Some cats are selectively bred so they do not cause allergies in people. Suggest 2 other reasons why people might selectively breed cats. Well, people may want to breed cats to either improve the well-being of the cats themselves or to improve their quality as pets. So for our answer, we're going to choose 1 point from each of these. So the first one could be to eradicate specific genetic disorders. This would be done by not allowing any of the cats with these disorders to reproduce. Now secondly, to improve cats as pets, we could breed them for aesthetic reasons. This could be the colour of their fur, the softness, for example. And finally, we could change their behaviour to make them better pets. This would be to make them less aggressive or more docile. You only have to mention 2 of these, and which 2 does not matter. Selective breeding could cause problems of inbreeding in cats. Describe 1 problem inbreeding causes. Well, inbreeding is where closely related individuals breed. This often occurs in artificial selection, when siblings are forced to breed because they have similar and desired traits. Now, when this occurs, it's much more likely for cats to pass on recessive disorders. Recessive disorders are determined by recessive genes. These are genes which are only expressed when 2 copies are present. That is when both the mother and father both have a copy of the gene. Now, in normal circumstances, this is very unlikely. But when siblings or very closely related animals interbreed, it is far more likely that both of them will have this recessive gene. So it's much more likely to be expressed and therefore cause disease. Alternatively, we could have phrased this in a different way and said the cats are more susceptible to disease. Many people have breathing problems because they are allergic to cats. This allergy is caused by a chemical called FeLD1. Different cats produce different amounts of FeLD1. A cat has been bred so that it does not produce FeLD1. The cat does not cause an allergic reaction. Explain how the cat has been produced using selective breeding. Now it's a 4-mark question, but it's level marked, and to get level 2, the top level, it needs to be detailed, coherent, and make logical links. Now the process of selective breeding is as follows. You choose the best cats for your desired characteristic. In this case, that is the cats with the least amount of FeLD1. You allow these cats to breed, and then from their offspring, you choose those with the lowest amount of FeLD1 production. And then once again, you breed them. So this is the process. And we need to explain how it happens. That is, describe what is done and also explain what is happening during these steps. Well, first of all, we're going to discuss what we've just mentioned. First of all, the cats with the desired characteristics, the lowest FeLD1, are chosen and allowed to breed. Our next point will explain what happens in this process. And this is that their offspring will produce varying amounts of the levels of FeLD1. Now of these, those with the lowest levels of FeLD1 will be chosen and allowed to breed with each other. It's important to remember that in selective breeding, like natural selection and any form of evolution, that it's a very slow process. So in order to see a visible change in the species, it is repeated over many generations. As a result, over time, the cats will produce less FeLD1, and thus will be suited for the purpose of not causing an allergic reaction. A student plans an investigation using mold. Mold spores are hazardous. Give one safety precaution the student should take when doing this investigation. Well, mold spores are tiny. Now this means they're airborne, so they are floating within the air. So the way they're toxic is that they are harmful to the lungs when breathed in. So to prevent this, we need to wear a mask, which will trap the spores and prevent them being breathed in. A student made the following hypothesis about the growth of mold. The higher the temperature, the faster the growth of mold. The student planned to measure the amount of mold growing on bread. The student used the following materials and equipment: slices of bread, sealable plastic bags, a knife, a chopping board, mold spores. Describe how the materials and equipment could be used to test this hypothesis. Now, this is a 4-mark question, and it's level marked. Now, to get to the top level, level 2, which is 3 or 4 marks, it needs to be detailed, coherent, and be logical. Now, in our investigation, our result should be quantitative, that is numerical, and the method should be repeatable, so that anyone could do your experiment and get the same results. So, using the equipment we have, we're going to expose some bread to mold and see how the rate of growth differs at different temperatures. Now, since we've been given a knife and chopping board, we can assume that we're going to not just take whole pieces of bread, but create exact sized pieces of bread, which makes the results comparable between the different temperatures. So that's the first thing we'll do. We'll take our piece of bread and we'll chop it up into 2 by 2 centimeter squares. This is a control variable, and it will be the same at all temperatures. We will then put some mold spores on each of these bread pieces. We'll then put 2 of these in each plastic bag, and these plastic bags we will expose to different temperatures. Each of these bags will be left at the given temperature for the same amount of time, and then the amount of mold on each will be seen at the end of this period of time. Now, the way I've described this experiment is only one way of doing it. There are multiple ways of carrying it out, and as long as the way you describe your method is detailed, coherent, and logical, and will allow the student to answer his hypothesis, you'll gain the marks. Okay, let's write out the method in a more detailed way to how I just described it. So first of all, take 4 slices of bread. By mentioning 4 here, instead of just saying take some bread, it shows the examiner we're being detailed. We're then going to say cut them into squares of side length 2 centimeters. After this, we're going to add 6 spores to each square. Place 2 of these squares in each plastic bag. Then place each bag at different temperatures. We're going to state which temperatures we're using, and 4 is a reasonable amount to use. So I'm going to say 20, 30, 40, and 50 degrees. We're going to say leave for 4 days. Measure the percentage cover of mold on each of the pieces of bread. Now, looking back at our answer, we can see that we've been detailed, such as saying that 4 pieces of bread and giving the exact temperatures. And we've also made it repeatable by talking about control variables. Give 1 variable the students should control in this investigation. Now, as you've already mentioned, it's very important that we control things in this experiment so it's repeatable. And we've already mentioned this in the previous part of the question. There were several things that we controlled. Now, one of this was the amount of mold on each bread piece. You could also say the size of pieces of bread, the types of mold, or the amount of moisture in each bag, because moisture affects the rate of growth of mold. Figure 3 shows the results. Let's have a look at figure 3. On the y-axis, we can see the amount of mold in units, whilst the x-axis shows time in days. Now, we have both 42 and 14 degrees here. Determine the rate of mold growth at 42 degrees between day 2 and day 7. Well, let's have a look back at figure 3. We are looking at the rate between day 2 and day 7, so we'll mark this on first of all. So rate is the change in something, in this case the amount of mold, divided by time. So, to find the rate of growth of mold, we're going to divide the change in the amount of mold by the number of days, which is the same here as doing the change in y divided by the change in x. Well, first of all, let's look at the change in y. Well, at day 2, there are 4 units of mold, and at day 7, there are 56. So the difference between them is 56 minus 4, which is 52. So that's our change in y. Our change in x is 7 minus 2, which is 5. Therefore, our rate is 52 divided by 5, which is 10.4. The growth of mold shows decomposition of the bread. Give a conclusion about decomposition from the results in figure 3. Well, in figure 3, at higher temperature, that is at 42 degrees, there is more mold since the line is higher. So, if mold is a type of decomposition, when there is increased temperature, there is also increased decomposition. Therefore, our conclusion is that the rate of decomposition is higher at higher temperatures. Figure 4 shows a reflex in the iris of the human eye in response to changes in light levels. Let's have a look at figure 4. Well, the most obvious difference here is the size of the pupil, the black circle in the center of the eye. Now, A is much smaller than B. Now, let's have a look at the first part of the question. Describe the changes in the pupil and iris going from A to B in figure 4. Explain how these changes occur and refer to the changes in light level in your answer. Now, it's a 4-mark question and it asks us to both describe and explain. So we want to aim to have 2 describe points and 2 explain points. Well, let's have a think about the structure of the eye. As we can see from this diagram, the eye is composed of 2 different types of muscles. First of all, the circular muscles, which are circles that surround the pupil, and the radial muscles, which are perpendicular to the circle muscles, and are like radii of a circle. Together, these muscles allow the eye to control the size of the pupil. Well, why is this useful? Well, it allows the eye to adjust how much light enters the eye and therefore reaches the retina. In high light conditions, such as on a sunny day, the eye will make the pupil small. This reduces the amount of light that enters retina to reduce harm to the retina. In low light conditions, such as at the end of the day or at night, the pupil will become as large as possible to let in as much of this low light conditions as possible to have the greatest chance of being able to see. Well, in A, the radial muscles are relaxed, which means they're elongated, so the pupil is smaller, whereas the circular muscles are contracted. When they're contracted, they become wider and therefore push apart, causing the pupil to become smaller. In B, the opposite is the case. The radial muscles contract, pulling together and shortening in length, therefore creating a larger pupil, and the circular muscles relax, becoming more thin. This causes the pupil to become wider, a process known as dilation, so the pupil is dilated in B. So let's construct our answer. First of all, we're going to state what has happened. So the pupils dilated at B. As it mentions in the question, we need to refer to the change in light level. So as it states in the question, this happened in low light levels. So that'll be our second point. Now we need to explain what causes this. Well, first of all, the radial muscles contract, and then the circular muscles relax. So let's have a look at the 4 marks. First of all, the pupils dilated at B gains 1 mark. Referring to low light levels is the second mark. The contraction of the radial muscles is the third mark. Relaxation of the circular muscles is the 4th mark. As we can see, the first 2 marks are descriptions of what is happening, and the 3rd and 4th marks are explanations of how this happens, giving a biological reason behind it. Some people wear glasses to improve their vision. Figure 5 shows light entering the eye in a person with blurred vision. Figure 6 shows how this condition is corrected with glasses. Well, let's have a look at figure 5 and 6. Well, as you can see with the rays of light in figure 5, they focus, seen by them crossing over, before the retina. Now the retina is the back of the eye, and it's what is sensitive to light. Light must focus exactly on this retina for a clear image to be seen. This is what happens in figure 6, and as we can see, the lens in the glasses bends the light so that eventually the light focuses on the retina. The question asks us to compare figure 5 and 6 and explain how the blurred vision is corrected. Well, as we've already said, the light does not focus on the retina, and since it focuses before the retina, it's known as myopia. Now this is what's commonly known as nearsightedness, where people can see near objects clearly but cannot see objects farther away. So this will be the first of our 2 marks. That figure 5 shows myopia, where light focuses before the retina. Now in comparison, as we've already said, in figure 6, light does focus on the retina, and this is because the lens bends the light. Since the light does focus on the retina, this will correct the blurred vision. So this is our second mark. So to summarise, in this question, we've both compared figure 5 and 6 by showing the differences in where light focuses, as well as discussing how the blurred vision is corrected. Two students investigated reflex action times. This is the method used. Student A sits with their elbow resting on the edge of the table. Student B holds a ruler with the bottom of the ruler level with the thumb of student A. Student B drops the ruler. Student A catches the ruler and records the distance as shown in figure 7. Steps 1 to 4 were then repeated. Well, figure 7 here shows the apparatus. We assume the ruler drops at a constant rate. Therefore, the length it drops can be used as a proxy for reaction time. The longer a person takes to realise that the ruler has been dropped, and therefore to clamp it in their hands, the further it will fall, and vice versa. Let's look at the first part of the question. Suggest 2 ways the student can improve this method to make sure that the test would give valid results. Well, if a test is to give valid results, it needs to be repeatable, meaning that anyone could do this test and get the same results. For it to be repeatable, we must have control variables. Well, let's have a look back at the method used. First of all, we see that the ruler is dropped. Now this could incur lots of differences when done in different ways. So this should be controlled. We can say they should be dropped from the same height. Now also a ruler is being dropped, and we need to ensure that we're using the same ruler, since different types of rulers may fall at different rates. And student A is catching this ruler, and we assume that one of their hands will be dominant, and will therefore be unfair if different hands are used. So they should use the same hand in each experiment. Finally, thinking back to the ruler being dropped, we need to ensure that it's dropped with no force, since if it was pushed down, it would move at a faster rate, and this could not be controlled between experiments. Dropping with no force means only gravity affects the speed of the ruler, which will be constant in all experiments. So for our answer, we can choose any of these. I'm going to choose being dropped from the same height and using the same type of ruler. But any could be used and would all get the marks. Table 3 shows student A's results. Now we can see here we have the test number in this column, and there's 7 tests, and also the distance the ruler is dropped in millimeters. What is the median result? And it gives us multiple tick boxes to choose from. Well, the median value is the middle value of an ordered list. So let's order this list of distances. from smallest to largest. Now, once we've done this, we need to identify which is the middle value. So there are 7 tests, 7 data points. So the middle value is calculated using n plus 1 over 2. So 7 plus 1 over 2, which is 4. So 4th is the middle. The 4th one is 117, which will be our answer here. The mean distance the ruler was dropped is 116 millimeters. Calculate the mean reaction time. Use the equation reaction time in seconds equals the square root of the mean drop distance in centimeters over 490. Give your answer to 3 significant figures. It's important to note the use of centimeters in the equation. So let's convert 116 millimeters, the drop distance, into centimeters, which is 11.6. This is because 10 millimeters equals 1 centimeters. Therefore, to go from millimeters to centimeters, we divide by 10. On the other hand, to go from centimeters to millimeters, we multiply by 10. So let's substitute 11.6 into the equation. We then get an answer of 0.1539. Now, we have to give our answer to 3 significant figures. Looking after the third non-zero digit, we can see a 9. Therefore, we should round up to 0.154. Now, let's look at where the marks come from here. Well, the first is for substituting the relevant values into the equation. The second is for the answer to this. And the third is for rounding to 3 significant figures. The students then measured student A's reaction time using a computer program. This is the method used. The computer shows a red box at the start. As soon as the box turns green, the student has to press a key on the keyboard as fast as possible. The test is repeated 5 times and a mean reaction time is displayed. Student A's mean reaction time was 110 milliseconds. Using a computer program to measure reaction times is likely to be more valid than the method using a dropped ruler. Give 2 reasons why. Well, a valid method means it's definitely repeatable. Now, there are some issues with using the ruler. First of all, is that somebody else is dropping the ruler. And humans are not machines. And therefore, you might be able to tell when the other person is about to drop the ruler. That's our first point. Secondly, to measure the distance dropped, you have to look at the rather large millimeter bars of the ruler, which is a very easy source of error. On the other hand, the computer will accurately determine the time to a millisecond. This makes the computer program more precise. Precision can be a difficult concept to understand. But if we imagine the exact same reaction time occurring 3 times, the computer is much more likely to show the same reaction time in all 3 than would be found from using the ruler test. A woman has a head injury. Her symptoms include finding it difficult to name familiar objects, not being able to remember recent events. Suggest which part of her brain has been damaged. Well, before we can identify which part has been damaged, we need to see what has changed. Since she cannot name familiar objects and is unable to remember recent events, we can infer that it's damaged her memory and her verbal reasoning skills. Thus, the part of the brain which controls these functions will be impaired. Let's think about the structure of the brain. Well, the largest part of the brain, the outer part, is known as cerebrum. Now, the outer layers of this are known as the cerebral cortex. And this deals with memory, intelligence, and language, and other things like this. Here we have the medulla, which is concerned with unconscious functions, such as breathing and heart rate. Next to this, we have the cerebellum, also known as the mini brain. Now, this controls coordination and movement. And finally, we have the hypothalamus, a small section of the brain that detects and controls the temperature and the water levels and is of vital importance for homeostasis. Now, this is a very simplified image of the brain. There are obviously other sections, but these are the main parts necessary for GCSE biology. So we need to look for the part of the brain that is concerned with memory and verbal or language skills. Now, as we've just stated, this is the cerebral cortex, so this will be our answer. A man has a head injury. He staggers and sways as he walks. Suggest which part of his brain has been damaged. Well, if someone is staggering and swaying, it suggests a lack of coordination, since they can't use their muscles correctly. So, let's have a look back to the previous part of the question and see which part of the brain controls this. Well, this is the cerebellum. So, for someone to lose coordination, there must be damage to the cerebellum. And that's our answer to this question. Figure 8 shows an image of a small section of DNA. Figure 9 also shows the structure of a small section of DNA. As you can see, Figure 8 shows the 3D structure of DNA. The structure we see here is known as the double helix. There are two strands of DNA curled together in a helical pattern. Figure 9 here is a more simplified version of the same thing. Now, we are asked what is part B. So let's have a look at one nucleotide. That's one of the monomers that make up the DNA polymer. Well, as you can see from this simplified version, we have a base, of which there are 4: adenine, cytosine, guanine, and thymine. And bonds between bases are what hold strands together. We have a deoxyribose sugar, which is a pentose sugar, meaning it has 5 carbons, which is why it's drawn as a pentagon. And bound to this, we also have a phosphate group. And together, these make a nucleotide. So, part B is a phosphate group. In Figure 8, the structure of DNA shows 4 different bases. There are 4 different bases, and they always pair up in the same pairs. Which bases pair up together? Well, as you've already mentioned, the 4 bases are adenine, cytosine, guanine, and thymine, also noted as A, C, G, and T. Now, of these, adenine always pairs with thymine, and cytosine with guanine. Now, we could write it like this, or we can use the letters, saying A with T and C with G. This question is a recall question, so that's sufficient for the marks. Figure 9 shows part of the gene coding for the enzyme IDUA. Well, let's have a look at Figure 9. The first one is strand J for a person without syndrome H, and the second is strand K for a person with syndrome H. Now, comparing the two, we can see they're exactly the same, apart from one base, this one here, where it's C on J and T on K. Well, strand K shows a mutation, which we've just identified, in the DNA, which has caused syndrome H. The enzyme IDUA helps to break down a carbohydrate in the human body. The enzyme IDUA produced from strand K will not work. Explain how the mutation could cause the enzyme not to work. So this is an explain question. We want to make 5 points. And in this we need to state what is happening, the mutation, and also explain what this causes in detail. Let's have a think about how an enzyme is produced from DNA. Well, DNA codes for a specific sequence of amino acids. These amino acids, in turn, come together to form a protein, in this case an enzyme. Now, the enzyme works by having a specific active site, which is complementary to the substrate, in this case the carbohydrate. Now, this active site is very sensitive, so any small changes will mean it won't work any longer. This is what's happened from the protein being made from strand K. So, let's discuss this. The change to the base will change the amino acid in the sequence. This will in turn change the shape of the protein. Now, since this is an enzyme, it's going to change the shape of the active site. Since the active site has changed shape, it will no longer be complementary to the substrate, hence it will no longer work. Okay, let's use this information to write our answer. Well, first of all, we need to describe the mutation that's happened. So the mutation has changed one base of DNA from C to T. This changes the amino acid coded for in the sequence. This could change the protein formed, so that the enzyme's active site was a different shape. So, the enzyme is no longer complementary to the substrate. Now, instead of saying substrate here, you could alternatively say carbohydrate. Now, this is sufficient for the full 5 marks. So, let's have a look at where those marking points were gained, with the red ticks shown here. A recessive allele causes syndrome H. A heterozygous woman and a homozygous recessive man want to have a child. Draw a Punnett square diagram to determine the probability of the child having syndrome H. Identify any children with syndrome H. Use the following symbols: capital A for dominant allele and lowercase a for recessive allele. Well, a recessive allele is one which it must be homozygous for the trait to be shown. Now, homozygous means that it's present on both chromosomes. Well, let's think about the genotypes of both parents. That is, which of the alleles each of them have. Well, the woman is heterozygous, meaning she has one of each allele. So we can refer to her as capital A, lowercase a. The man is homozygous recessive, so has 2 recessive alleles, noted by 2 lowercase a's. Using this information, let's draw a Punnett square, as follows. Now, the 2 symbols that you can see are the scientific symbols for male and female. The top one being for females, and the one on the side for males. A Punnett square is used because of the 2 alleles each parent have, their offspring, that is their children, will receive only 1. So, in this case, each of the 4 boxes here represents a possible child, and they will receive 1 allele from each of their parents. So let's fill it in, using this logic. We can see that 2 of the possible children will be capital A, lowercase a, which is 1 dominant, 1 recessive, known as heterozygous, and the other 2 will be 2 lowercase a's, meaning they have 2 recessive alleles, known as homozygous recessive. From what we discussed at the start of this question, we said that anyone with 2 recessive alleles will inherit the disease. So, 2 of the children, both of the 2 lowercase a's, will have the syndrome. So, the probability of the child having syndrome H is 2 divided by 4. That is, 2 of the possible children have the syndrome divided by 4, the total number of children possible. Now this equals 0.5. You can also express the probability as a percentage, which would be 50%, or as a fraction, 1/2. I'm going to use 0.5, but any of these are valid. Food security is when a population has enough food to stay healthy. Lack of food security is a global problem. One way to maintain food security is to increase the efficiency of food production. Figure 10 shows how some pigs are farmed using intensive methods. Well, as we can see from Figure 10, there are lots of pigs in a crammed environment. There are bars to keep them within a cage, as well as food readily available in this trough. Well, what does the question ask? Some people think the farming methods shown in Figure 10 are unethical, suggest 2 other possible disadvantages of intensive farming methods. Well, as we just discussed, they're very close together. This makes it very likely for the disease to spread between them, which is a large issue. Also, places such as this, where intensive farming occurs, are often heated. This requires the use of fossil fuels, which presents problems. So, the 2 issues are that diseases spread more rapidly, and that the heating requires an increased use of fossil fuels. Alternatively, you could just discuss antibiotics. Due to the potential of disease spreading, farmers often use large quantities of antibiotics, so they are often overused, and this could cause resistance to antibiotics, which poses large issues for the agricultural industry. Explain how the intensive farming of pigs increases the efficiency of food production. It's a 4-mark question and it's level marked. And to get level 2, 3 or 4 marks, we need to make clear statements which are linked to relevant explanations. Now we want to aim to have at least 2 of each of these. Well, let's think about what efficiency means. Efficiency is essentially the proportion of energy used which is converted into meat. When we're talking about energy here, we generally mean chemical energy from the food that animals eat. As we touched on earlier, the animals are kept in a temperature-controlled environment, as in they are heated. Now, since this happens, they must use less energy to heat themselves, since pigs, like all mammals, are warm-blooded. And less energy is transferred to the environment, which happens when organisms heat themselves through respiration. Next, we're going to talk about their lack of movement. Now, as we said earlier, their movement is restricted since they're kept in cages. Now, this reduces the amount of energy that's used for movement. Movement requires muscle contraction, which uses energy from respiration. So, with less movement, less energy is wasted, so more energy is available for growth. Let's look back at our answer. We can see here we have a statement followed by a clear and relevant explanation, and this is repeated in our second point. Although it's level marked, the following red ticks roughly show where the marks will be gained. A newspaper reported that food security is a serious problem in remote communities in Canada. This is because Aboriginal communities are eating fewer traditional foods. One traditional food eaten by Aboriginal communities in Canada is seal. Look at Table 4. Well, Table 4 shows in one column the year, from 2004 to 2010, and in the other column the number of seals caught in thousands. And we can see a clear trend in decreasing down the years. Calculate the percentage decrease in the number of seals caught from 2004 to 2010. Well, percentage change, in this case a decrease, is calculated using the formula final value minus initial value divided by the initial value multiplied by 100. So let's identify the values we need to plug into this formula. In 2004, there were 362,000, and in 2010, there were 67,000. So our final value is 67, and our initial is 362. Now, although these values are in thousands, we can use them as they are, since percentage change shows change as a proportion of the initial value. So this proportion will be the same, regardless of whether these values are multiplied by 1,000. So, plugging it into the formula, we get 67 minus 362 divided by 362 times 100 equals minus 81.49. Now, the negative value is due to the fact that this is a decrease. Now, we're going to give our answer to 3 significant figures, so 81.5. We do this to be safe, as 3 significant figures will be accepted by the mark scheme where the question does not state a specific number of significant figures. The conclusion in the newspaper might not be correct. Suggest 2 reasons why. Well, the newspaper suggested that the food insecurity in Canada in Aboriginal communities was due to less traditional food being eaten. Well, we don't know what else they eat. Their diet might not be entirely reliant on traditional foods. So this is our first point. They may eat other foods, so it's unclear if they're food insecure. Also, we are only given information about seals, and we do not know if other traditional foods have declined. Therefore, there's not enough information to make a conclusion based off just one type of food. This is our second point. Homeostasis controls the internal conditions of the body. Explain how blood glucose levels are controlled in the body of someone who does not have diabetes. Well, let's think about how blood glucose levels are controlled. Well, initially, we have normal conditions of blood glucose. But what happens if they change? Well, first of all, if there's an increase in glucose concentration of the blood, then insulin is produced. This is a hormone that travels through the blood and is absorbed by receptor cells. It causes cells to absorb glucose, and it also causes the liver to take up glucose and turn it into glycogen. This is a storage carbohydrate. It's similar to starch in plants. Now, these processes take glucose out of the blood, thus reducing the blood glucose levels back to normal. Well, let's think about what happens in another case, when the glucose levels become too low. Well, in order to increase the glucose concentration of the blood, glucagon is released. Now, this is a hormone, and the hormone stimulates the liver to convert glycogen that we previously discussed into glucose, which is subsequently released into the blood. This will therefore increase the glucose level back to normal. So let's write this into our answer. First of all, if they are too high, insulin is released from the pancreas. So glucose moves into cells. If too low, glucagon released from the pancreas. So glycogen converted into glucose and released into the blood. Now, for this final mark, you must say both that
Let's have a look at where the marks are awarded
and that it's released into the blood.
The first one is about the release of insulin.
The second is about glucose moving into cells. We then get another mark for discussing the response to low glucose, which is the release of glucagon. And finally, we get a mark for saying about the conversion of glycogen into glucose and the release into the blood of this glucose. Compare how each type of diabetes is caused. Suggest how each type of diabetes can be treated. Now, since it says compare in the statement, we must make some comparative statements using comparative language. So let's think about each type of diabetes. Type 1 is one that you are born with, and it is where your body produces no or insufficient insulin, whereas type 2 is an insensitivity to insulin due to genetic and environmental factors. So let's first of all compare the causes of each. So in type 1, not enough insulin is produced, whereas in type 2, cells do not respond to insulin. By using whereas here, we are making it comparative. Let's think about the next part of the question and how each of them can be treated. Well, due to the nature of type 1, there is a physical lack of insulin. This can be treated by injecting insulin into the body. On the other hand, there is still insulin in the body of those with type 2, but the cells do not respond to it. Since it's caused partly by environmental factors can also reverse it. Therefore, it is treated with diet and exercise. By regulating these, the blood glucose levels of individuals is reduced, and over time, the sensitivity to insulin increases. These are our third and fourth points. So let's look at how the marks are awarded. We get 1 mark each for describing the cause of each type of diabetes, and the third and fourth mark come from each of the respective treatments for the types of diabetes. Look at Table 5. Well, Table 5 shows the population of the UK in 2015, the number of people diagnosed with diabetes, and the estimated number of people with undiagnosed diabetes. The figures are given in standard form. Calculate the percentage of the UK population estimated to have diabetes. You should include both diagnosed and undiagnosed people in your calculation. Give your answer to 2 significant figures. Let's think about what we're trying to find. Well, the percentage of the population with diabetes is the same as the number with diabetes divided by the total population multiplied by 100. Now to find this, we need to first of all work out the total number of people with diabetes. And this is the sum
of those with diabetes that are diagnosed and those that are estimated and undiagnosed. So adding those together, and we can do this with standard form using our calculators, gives us an answer of 3.999 times 10 to the 6. We can then substitute this into the formula we just discussed. 3.999 times 10 to the 6 divided by 6.5 times 10 to the 7, that is the total population, multiplied by 100, gives us an answer of 6.15. Now the question asks us to give our answer to two significant figures. So looking after the second digit, we can see that it's a 5. So we should round up to 6.2%. Now, we only get 1 mark for this final answer. The other marks come first of all from working out the total number of people with diabetes. The second comes from working out percentage, and the final one is for rounding to two significant figures. A urine test can be used to check for the presence of glucose in the urine. Diabetes can also be diagnosed with a blood test to measure the concentration of blood glucose. Suggest why a blood test is more reliable than a urine test. Well, there are several reasons for this. The most pressing of these, perhaps, is that of timing. The blood test gives a current result, the immediate amount of glucose in the blood, whereas urine levels may be hours old. As in, the urine produced from the blood will have been produced up to hours ago. Therefore, it does not immediately reflect the amount of glucose in the blood, which the measurement of urine is a proxy for. We could also say that it's also more reliable since there's not always glucose in the urine of those with diabetes. Now, finally, we could also say that glucose in the urine could be due to other reasons, such as kidney disease, which affects the amount of glucose reabsorbed by the kidneys, and hence blood glucose levels. A blood test, called the glucose tolerance test, checks how well the body processes glucose. Concentrations of glucose in the blood are measured before and after drinking a glucose drink. Patients are not allowed to eat food for 8 hours before the glucose tolerance test. Suggest why patients are not allowed to eat for 8 hours before the test. Well, food generally contains glucose. So, glucose from the food would affect the result. Therefore, leaving a long amount of time prior to the test means that the insulin in the blood will act on the glucose from the food, thus taking it into cells and the liver so that blood glucose levels are reduced. Now this is our answer. Alternatively, we could discuss how leaving 8 hours means that there's a low starting point for glucose. This means it would show the effects of the glucose drink more clearly. Alternatively, you could say so the results are not affected by the glucose from food. Any of these are acceptable. Figure 11 shows the results of a glucose tolerance test for two patients, A and B. Let's have a look at figure 11. We can see on the y-axis we have blood glucose concentration in units, and on the x-axis we have the time in hours. Now at the start of this, a glucose drink is taken at 0 hours, and we have the response of two different patients to this drink. Where, as we can see, patient B shows an increase and a dip, whereas patient A shows a constant increase throughout the time, as well as having a greater level of glucose throughout. The question asks us, which patient has diabetes? And it asks us also to justify your answer. Well, let's have a look back at the figure. So, with someone without diabetes, we would expect an initial rise in glucose due to the drink, as seen in patient B. Then insulin would begin to be produced and released into the blood, which would act to lower blood glucose levels since it's absorbed by cells in the liver. On the other hand, in someone with diabetes, there's either not enough insulin or an insensitivity to this insulin. Therefore, the blood glucose levels will not fall. Due to this lack of insulin, or resistance to it, we would expect someone with diabetes to also have a higher blood glucose level throughout. This can be seen from patient A. Therefore, patient A has diabetes. So that's our answer, patient A. And the reason for this, and you get 2 marks here for the justification, are that the glucose levels are much higher and they remains high. So, as we previously discussed, both of the marks come from the justification, from stating that the glucose levels are higher and that they remain high. Endocrine glands produce hormones. Hypothyroidism is caused by an overactive thyroid gland. Suggest what would happen in the body of a person with hypothyroidism. Well, let's think about what the thyroid gland does. Well, the thyroid gland produces thyroxine, and thyroxine controls the metabolic rate of the body. So, when it says suggest what happens in the body, it means physiological processes, such as chemical reactions, etc., rather than symptoms that one with this disease would experience. So, let's write our answer. So first of all, we're going to state what's happening, an increased production of thyroxine, and then what this causes. Therefore, greater BMR, which stands for basal metabolic rate, the base rate of metabolism without exercise. So essentially, the amount of chemical processes going on at rest. So therefore, there's an increased rate of respiration. Now for this third point, you can discuss any other metabolic process and talk about an increase, such as an increased rate of protein production, for example. So, in this question, the marks come first of all from talking about more thyroxine, secondly from a greater BMR, and finally from an increased rate of respiration or other metabolic process. Describe the roles of FSH and LH in the menstrual cycle. Let's think about each of these hormones in turn. Well, first of all, FSH. This stands for follicle stimulating hormone. Now, it's produced in the pituitary gland, in the head, and its main role is to cause the egg to mature. It also stimulates the ovaries to produce estrogen. Now let's think about LH. LH stands for luteinizing hormone, and LH is also produced in the pituitary gland. It is released in large amounts just before day 14, and it results in the release of an egg, and thus ovulation. So let's write this in our answer. First of all, FSH causes the egg to mature and stimulates the ovaries to produce estrogen. LH causes the release of egg at ovulation. Now, the marking in this question is 1 mark for each of these statements, as seen here with red ticks. The combined pill is a contraceptive that contains progesterone and estrogen. The mini pill is a contraceptive that only contains the progesterone hormone. It has to be taken at the same time each day to prevent pregnancy. The success rate of the mini pill in preventing pregnancy is lower than that of the combined pill. Explain why missing a dose of the mini pill would reduce the success rate of the mini pill. Well, let's think about what this mini pill does. Progesterone is a hormone which inhibits FSH and LH. These are essential for the maturation and release of an egg and therefore pregnancy. So the inhibition of them prevents the possibility of pregnancy. It has to be taken at the same time each day so that it maintains a constant or similar level in the bloodstream, which is high enough to inhibit these hormones. So let's look at this graphically. So if we look at a graph of the amount of progesterone in the blood and days on the x-axis, we can see that the level will fluctuate, but it will fluctuate around a similar value. At the point at which the pill is taken, the level of progesterone is on the lowest. It then increases as it's absorbed into the body and decreases as it's broken down. However, if it is not taken at one point, it will continue to decrease. Therefore, the level of progesterone in the blood will be far too low to inhibit the FSH and LH appropriately. If this were to happen, then FSH and LH would act normally. In this way, FSH would cause the maturation of the egg, and LSH would release it. So let's write this into an answer. Missing a dose causes a drop in the levels of progesterone in the blood. FSH is no longer inhibited. This drop means that LH is also inhibited. Finally, we need to discuss what this will cause to happen. So, consequently, the egg will be matured and released. And this is what can cause the possibility of pregnancy. Let's have a look at how the marks are awarded here. Firstly, for mentioning a drop in progesterone, then a mark each for FSH and LH no longer being inhibited, and finally for what this causes, that the egg may be matured and released.